In order for you guys to be able to do a good job on your lab report, I want to let you know about a few details that you might not be aware of. The first thing is that we actually used a GFP variant in this lab prior to performing mutagenesis. That means that our GFP is not exactly the same as GFP Wild Type. It has 3 mutations. It has a serine to a glycine at position 65. It has a serine to an alanine at position 72. And it has a glycine to an alanine at position 232. Now, the one that's the most interesting is the serine to the glycine at position 65. Why? Because that is smack dab in the middle of the chromophore. Interestingly, the wild type fluoresces more brightly than our variant. They both fluoresce green, and they're both quite visible, but the wild type is in fact substantially brighter. So you want to keep the chromophore in mind generally when you're talking about your lab report, and you're also going to want to keep in mind that you might even have a mutation to the chromophore itself. One way to learn more about GFP is to go to an excellent website called Uniprot. It's actually a database, and it has information about all kinds of proteins. It has a great deal of information about our green fluorescent protein. So I would strongly recommend Googling GFP Uniprot, finding this web page, and just reading through it and learning a bit more about this protein. This is a great source of information. Not only does it have information about the function of GFP and many other aspects of it, but it also has detailed analysis of the mutagenesis work that has been done on GFP and how different variants have been developed that fluoresce different colors. Just as an example of that, if you look at this very top example of mutagenesis, serine, tyrosine going to a threonine tryptophan actually gives rise to cerulean, which is a kind of a blue. You can think of like the cerulean sky type of fluorescence. And if you go on down, there's just many, many, many examples of this. It's pretty amazing. So in your analysis of your mutant, it is possible that you might have a frame shift. This happens to be a little more tricky to analyze than a point mutation or a silent mutation. So what you're going to want to look for is a dash. There are 2 possibilities. You could have a deletion in your mutated sequence, or you could have an insertion. Here we actually see the insertion of an A. Whether it's a deletion or an insertion, it doesn't really matter. It's going to throw your reading frame off. And you will notice if you go down here and look at the amino acid alignment that your amino acids are aligned quite nicely until you hit the frame shift, and then all of a sudden your sequence is radically different from GFP. And that is because the reading frame has been shifted and the amino acids that it would translate are completely different. Another thing that you might run across is a mutation that we call a nonsense mutation. It is a single nucleotide change, but it's the case where it leads to the insertion of a stop codon. And if you notice here, we have a G to a T. And what was the end result of that? Well, we end up with a stop codon. TAG, as you know, is a stop codon. So you can imagine that if you have a stop codon inserted in the middle of your protein, that's not going to help it very much. It's probably going to destroy whatever function the protein may have, particularly when it happens very early or in the middle of the protein. Here, it's happening at position 124. The total length of GFP is 238 amino acids, so this would be very likely to have a white phenotype because the GFP would not be functional at all. So I want to clue you in about a handy little tool in Eugene, which is exporting your alignment as an image. So basically you can go to the menu or you can right-click anywhere in the white space, you choose Export, and then you choose Export as Image. This will bring up a window, and I like to include the sequence name, so I check that box. It goes to a folder in your documents. It goes to a Eugene data folder in your documents. You could modify that, but I don't really mind it. I just leave it as is. Hit export, but then you have to go find it. It's not going to pop up. You have to go to that folder and actually physically find it. Since we're on the subject of images, I would like to talk about how to present your gel, whether it's SDS page or agarose gel, correctly. The first thing you need to do is make sure you have labeled all your lanes. This begins with putting an M where your ladder is and then numbering all your experimental samples. You should also definitely label your marker or your ladder, whatever you like to call it, with the appropriate molecular weights in the case of SDS page, or base pairs or kilobase pairs in the case of a DNA gel. The other thing you can do, which I think is very useful, is if there's something you want to highlight, you can use an arrow or you can use a little box to circle a band. You don't have to be shy about modifying your image. The other thing that's very critical is to place a figure legend below the gel. That figure legend must have a brief descriptive title, and then you need to provide a clear description of each lane. Making your figure complete and well done is a really great way to impress your TA and get a good grade on your lab report.